Good morning. This presentation is based on the applications of plant tissue culture. And this is for the fourth semester B.Pharm students under the third chapter of plant tissue culture. Already we have seen in this chapter regarding your plant tissue culture, its introduction, history, we have seen. And now we will see about the applications of plant tissue culture in this presentation, and the different types we will see in the next presentation. So already you know that plant tissue culture is nothing but growing of an isolated plant tissue or a cell in an artificial medium by providing the conditions, all the required environmental conditions to develop into a whole intact plant or to produce the secondary metabolites, whatever you decide. So in that context, if you see the applications, that means what are the uses of this plant tissue culture? So when you just see the applications of plant tissue culture, it is playing its important role in different fields. That is in the first field, that is your agronomical field, and another one is the industrial applications. In the agronomical applications, we can see the rapid multiplication of selected plants by the multiple production of plants identical to the original one. And new plant is also obtained from, which is different from the original and also with the more efficient criterias. Maybe due to the, or maybe the high yielding or herbicidal capacity or the drought resistant plant or the plants which can grow in the drought or insect resistant plants or sometimes the salt resistant crops. And when you just see to the industrial applications, mainly it depends on the production of secondary metabolites by using the biosynthetic capacities of plant cells, which are allowed to grow in a bioreactor. That is nothing but the fermenter, otherwise you can call it, or otherwise the situation where you are providing the artificial environmental condition in the nutrient medium. And also in the industrial area, it can also find its application in the innovative aspects, that is by employing the new source of variability accessible in vitro to obtain the new molecules. And that is very crudely in two different ways the plant tissue culture is useful. And now we will see what are the specific applications of this plant tissue culture method. Plant tissue culture technology has been used in almost all the fields of biosciences, and its applications includes the following things. The very first application is production of phytopharmaceuticals and secondary metabolites. And this secondary metabolites or phytopharmaceuticals can be produced by many ways. One is by biotransformation, that is otherwise we can call it as biochemical conversion. And second one is by plant cell immobilization. And third one is by genetic transformation, that is nothing but the production of transgenic plants, and also the production of compounds by using the bioelicitats. And the second application of this plant tissue culture is your micro propagation, that is nothing but clonal propagation. And next one is production of the synthetic seed, production of protoplast culture and somatic hybridization. And next is hairy root culture, then Creo Preservation, and tracing the biosynthetic pathways of secondary metabolites, and generation of novel compound from the plants. The various plant organelle functions can be studied, like respiration, organ function, and metabolism in the plant. And plant improvement studies also can be done to produce the disease free plant by studying the various kinds of diseases occurring to the plant. And also mutant cell selection is done by this plant tissue culture method, that is by adding the toxic substance to the cells during the production, and then by isolating the resistant cells. Thereby you can study the mutant cell selection. And also production of highly economical valuable substances by the plant tissue culture can be done, because for the subjects or for the components where they don't have any other chemical methods for their synthesis. So that's how it is highly valuable in production of the economical valuable compounds by this technique. So these are all the various applications I have listed out, around 12 applications of this plant tissue culture. And now we will see one by one how it is important. The first application is production of phytopharmaceuticals and secondary metabolites. So the secondary metabolites like alkaloids, terpenoids, flavonoids, or the primary metabolites like your lipids and oils, and the secondary metabolites again like your tannins, anthraquinones, flavones, naphthoquinones, vitamins, and the other substance, protein derived substances and proteins, and anti-cancer agents, antiviral agents are produced by this plant tissue culture. So next I'll just show you the table where the different compounds from the which plant species and what is the type of culture it is used to produce. You can see the first one, the anthraquinones, which is the plant species used here is Acacia angustifolia through callus culture. This can be obtained, and caffeine from Coffea arabica by callus, and cardinolites, that is digitoxin, digitoxygenin, gitoxin from Digitalis purpurea, both by suspension and callus culture, and codeine from Papaver somniferum through suspension culture, and diasgenin from Diascorea composita from callus, and again glycyrrhizin from Glycyrrhiza glabra through the suspension culture, and pepine from Carica papaya from callus, reserpin from Rauwolfia serpentina from the suspension culture. And also there are other examples I have given, what are the component and the plant species used and the type of cell culture used. And next one is the biotransformation. The plant tissue culture is also having its importance in the production of secondary metabolites through biotransformation. What do you mean by that biotransformation? It is a process in which the functional group of organic compounds are modified by living cells. So this biotransformation may be of single step or may be of double step. So the precursor, what you are using, is otherwise called as your substrate. And then you will be adding some of the substance, that is the chemical, which converts the substrate into the product through a living cell. And this process is done by using a microorganisms, or sometimes the plant cell in a suspension, or sometimes hairy root culture or immobilized cells can be used for this conversion. The biotransformation by plant cell culture is brought about by various types of reactions like glycosylation or glucosyl esterification, hydroxylation, oxidoreductions, hydrolysis, epoxidation, isomerization, methylation, demethylation, or dehydrogenation, etc. It is not only employed to increase the yield, but also it is highly economical for the commercial production. And here you can see some of the examples of the secondary metabolite production by the conversions. The very first class of the compound is alkaloid. Here the substrate used is codenone, which is converted into codeine by single step process in Papaver somniferum species. And the example of phenolics, resorcinol is converted into glucoside in Datura inoxia. In case of steroids, digitoxin is converted into digoxin. In case of digitalis lanata. And the phenolic flavonoids, liquidity-genin is converted into glucoside in case of Datura inoxia or Catharanthus roseus. And phenolic anthraquinones, that is rain, is converted into rain glucoside by in the species Perilla fructescens. So other two examples are also given, coumarins and terpenoids, where the substrate is converted into product by single step in a specific plant species. The next one is by plant cell immobilization. What do you, before going into the applications, that what first you should know, what do you mean by the cell immobilization? Already I just told you about the cell immobilization. That is nothing but it is a technique in which the catalytically active enzyme or a cell within the reactors or a system, it is made static so that its entry into the mobile phase can be reduced. Mobile phase is nothing but your liquid medium or the nutrient medium where only the substrate and the product will be present, whereas the enzyme or a cell which has to act on that substrate to convert into product is present in a static manner. So this immobilization involves the entrapment of cells within a gel or a passive adsorption on solid support material. So when you just take the production in the plant tissue culture, how it is happening, the substrate and the product is present in the liquid medium which are freely moving, and the immobilized non-growing cells or the enzymes are present in the solid matrix within the medium. It is a controlled process of agglutination or formation and adhesion on a matrix under the controlled condition. It is a process in which the highly reactive and sensitive enzyme is embedded or trapped in a matrix so that only the substance and product can pass through the matrix. And the cells are cease to grow and accumulate the metabolites. In immobilization technique, the plant cells or the enzymes are entrapped in different polymerized matrices. The example may be alginate or agar or agarose, etc. And that can be converted into an heterogeneous catalyst. And there are different types of immobilization. They are like your adsorption, which is nothing but the direct intracellular binding due to natural affinity. And second one is your cross-linking, that is covalent coupling on inert matrix, and cross-linking, that is intracellular connection via bi or polyfunctional reagents. And third one is embedding, that is mixing with suitable materials by changing their consistency with the temperature. And the last one is the entrapment, the physical retention within the framework of diverse pore size and permeability, that is micro encapsulation. That means you are allowing the cell or an enzyme to get entrapped itself within a solid matrix. So in that, first when you just see what do you mean by entrapment, the gel entrapment by polymerization with the polymers like polyacrylamide, example Vinca. And second one is gel entrapment by ionic network formation, that is the entrapment of cell in calcium alginate. Usually we do this method in our laboratory. First what we will do is we will prepare the sodium alginate solution, and with that we will mix the yeast cell suspension. That means here we are entrapping the yeast cell by this gel entrapment method. And once you mix this sodium alginate and yeast cell, and that one is made to entrap as a gel in calcium chloride solution. So what happens? The sodium gets replaced by calcium and it gets stabilized. Okay, and the cell gets entrapped within the calcium alginate matrix. And then the activity of the cell is determined by either subjecting it into the hydrogen peroxide solution and incubating in the incubator for specified period of time, which shows the respiration activities, or otherwise by the action of enzyme present in the yeast, zymase, on the sucrose solution, and where you can find the reduction activity by Fillings test. That usually we do for the fourth year students, but I don't think the semester students have that demonstration or the experiment in your practicals. And the gel entrapment by precipitation of some natural and synthetic polymer by changing one or more parameters such as pH, temperature, and salinity. And also entrapment is performed in the hollow fiber reactors, that is called as your polyurethane foams. And the other type is surface immobilization, that is with the help of nylon, cellulose, etc. that is done. And again, the immobilization can be done by embedding in agar and agarose beds. And when you just see the applications of this plant cell immobilization, that is the through biotransformation, the secondary metabolites can be produced via cell immobilization, and rapid biosynthesis of secondary metabolites can be done, and synthesis from the precursors also can be done, and the biosynthesis of secondary metabolites with increased yield over extended period of time can be achieved. So here I had given some examples where the secondary metabolites can be produced from the plant species like capsicum, coffee, vinca, mentha, tobacco, etc. And the next application is the genetic transformation, that is the production of the transgenic plants. What do you mean by this transgenic plant? The plants are produced through genetic engineering methods, that is the transfer of gene from an unrelated organism to the related one. Such genes are called transgenes. And the plants containing transgenes are called as transgenic plants. In other words, the genetic information can be defined as the transfer of foreign genes or the recombinant genes isolated from plants, virus or bacteria into a new genetic background. Already, this foreign gene or a recombinant DNA is present in the plant or the smaller microorganisms that can be transferred to a new genetic background is called as your genetic transformation. The targeted cells for gene transformation are cultured cells or protoplast, or sometimes meristem cells from the embryos, or pollens or zygotes, or sometimes cells from immature embryos, or cells from shoots or flowers. That means the foreign genes or can be transferred to these kind of cells. That is nothing but your cultured cells or protoplast or meristem cells from the embryos or pollens or zygote, or immature cells of embryo, shoots or flowers. What are the applications of this transgenic plants? When you just transfer the foreign gene to the various targeted cells, you will result in the plants with the different characters. So what are that different characters we come across, or the applications of transgenic plants? So here what happens, whatever the gene you want to incorporate, that can be done. That is the first application. That means that is nothing but genes have been successfully transferred to many crops for resistance to various biotic stress. And gene resistant to abiotic stresses like herbicide resistance plants can be obtained. The resistance against viral infection can be brought about. This gene transfers also improves the quality of food products. And also it is helpful in male sterility and fertility restoration in transgenic plants. The transgenic plants will have both basic and applied role in the crop improvement. So here I have given the examples of plants where this transgenic plants has been obtained. The example are tobacco, tomato, soya bean, shatavari, papaya, licorice, neem, etc. And next one is the production of the secondary metabolites or the wanted traits by the elicitors. So you should know, what do you mean by this elicitor? Elicitor is nothing but any substance that may be an organic substance or an inorganic agent which is used to trigger or stimulate the rapid and increased production of secondary metabolites, or sometimes the rapid growth of plant cell culture, or organs. That is called as the elicitors. Elicitors are nothing but they are the agents that may be of organic nature or inorganic nature, which stimulates the production of secondary metabolites or stimulates the growth of the plant cell in the culture. How this stimulation is happening? That stimulation can be brought about by induction of stress in the plant cultures in terms of specific environmental, physiological, or biological conditions. That means that stimulation is brought about in changing the environmental condition or physiological condition or biological condition. When you just change any of these things, naturally the cells will have more stress and they get stimulated to produce more amount of secondary metabolites. This process is called as elicitation. That is the production of more amount of secondary metabolites or increase in the growth due to the stress by the changes in environment, physiology, or biological conditions. The secondary metabolites can be synthesized and accumulated to this response are called as phytoalexins. And they also act as the defense agents to invading pathogens. The signals which triggers the formation of phytoalexins are called as elicitors. So now you got the point. Any organic or inorganic compound which is used to trigger or stimulate the production in an enormous increased way or the increased growth of the cell in the culture is called as your elicitor. And the products which are synthesized or which are produced in large amount due to this stress are called as phytoalexins. Okay, and any signal or any agent which induce the formation of phytoalexins are called as elicitors. Okay, and the classification of bioelicitors can be done by two bases. One is based on the origin and another one is based on the nature. Based on the origin, there are two types of elicitors. One is the exogenous elicitors and another one is endogenous elicitors. Exogenous are nothing but the compounds which are outside the cell, maybe the enzyme, metal ions, UV light, phytosan, etc. And endogenous, that is the enzymes which are present inside, hepta, beta glucoside, or the substance which is present inside, dodeca beta 1,4 D-galacronide. And next, the classification of elicitors based on the nature, that is biotic elicitor and abiotic elicitors. So first you should know what do you mean by the biotic elicitors. That is, they are derived from the microorganisms or produced within the plant cell by plant defense process. That is, when a plant gets infected with the microbial infection, they will produce some defense products. They are called as the biotic elicitors. That is the antibodies, especially when you say in case of our animal tissue, the antibodies. When an antigen enters, it produces the cells against that antigen to fight. That is, other ways they are the antibodies. So likewise, when when the plant is getting infected with the microbial infections, they produce some of the substances within themselves to fight against that microbial infection. They are called as biotic elicitors. So the beta-linked glucans, chitosan, enzymes, cell wall derived polysaccharides like pectin, pectic acid, cellulose, etc. are the biotic elicitors. And these are added to the medium in low concentration, that is around 50 to 250 nanogram per liter, to enhance the metabolite production. And next one is the abiotic elicitors. The product accumulation is due to the stress caused by the physical or chemical agents, that is external agents like your UV, low or high temperature, or antibiotics, salts of heavy metals, and the temperature conditions like freezing and thawing, and the non-essential components of the media like agarose, tin, and agaropectin, and some of the chemicals like that is methyl jasmonate. It is nothing but the plant growth regulator, and copper sulfate, silver nitrate, or high salt concentration are considered to be the abiotic elicitors. Abiotic elicitors means these are the substances, either physical agents or chemical agents, which induce the production apart from the things which are present outside the cell. That is called as abiotic elicitors. And generally, the elicitors stimulate the accumulation of secondary metabolites. So there are some examples in case of opium, diascorea, datura, vinca, capsicum, carrot. The secondary metabolite production is increased by these elicitors. And next one is the micro propagation, which is otherwise called as clonal propagation. It is nothing but it is the ability to regenerate the plants directly from the explants or from a single individual asexual cell into a clone. That means the single isolated cell or a isolated tissue will propagate. That means it will multiply in the enormous number with the same kind of characters. That is called as your clone. It is defined as a type of propagation of selected genotypes in vitro culture techniques. That is especially in case of vegetative method of propagation of plants. The micro propagation or cloning of tissue culture or growing of cells by the in vitro method. Okay, that is outside the natural environment. You are providing everything and you are developing the multiples of the original cell, what you are using it as an explant, is called as your micro propagation. And in this, what are all the things you can develop, or how this micro propagation is useful in the vegetative propagation? If you see, micro propagation by proliferation of axillary buds and adventitious shoot proliferation can be brought about. Production of artificial seeds and plant regeneration by organogenesis, that may be either from callus or from the suspension. And same the plant regeneration, either from the callus or suspension by somatic embryogenesis, or sometimes the direct somatic embryogenesis can be brought about. These are the ways in which the micropropagation is playing its role. Okay, so the methods of micropropagation, that is already we have seen. I have told you in the last slide itself, that is micropropagation by the proliferation of axillary buds or adventitious shoot proliferation, or by artificial seed method, and plant regeneration by organogenesis from callus and suspension, and plant regeneration either from callus or suspension by somatic embryogenesis, or otherwise by direct somatic embryogenesis. So what are the advantages of this micropropagation? Here it is rapid multiplication of superior clones can be maintained, and we can maintain the genetic uniformity or high yielding crops, or whatever the desirable characters can be maintained within a very short time period. And also we can maintain the multiplication of sexually derived sterile hybrids, and also improve the sexual characters of the plants. We can also able to develop the virus-free, insect-resistant or disease-resistant or herbicide-resistant plants. That is, in the other words, what we can say is the unwanted or undesirable characters can be removed by this micropropagation techniques. Example, that is, when you just take fennel, it is genetically heterozygous in nature, and we can see the variation in oil yield and composition under this micropropagation. And also we can bring about the many characters in the production, as well as increasing the production of secondary metabolites in the various plants like garlic, brahmi, vinca, eucalyptus, gymnema, liquorice, etc. And next one is nothing but the production of the synthetic seeds. That is the this area, the micropropagation is also helpful in the production of synthetic seeds. That is the artificial seed, or otherwise called as somatic seed or synods or synseed. There are many names for the synthetic seeds. What do you mean by that synthetic seed? They are the artificially encapsulated somatic embryos, or the shoot buds or cell aggregates, or any other tissue. They are used for sowing. They have the ability to convert into a plant under in vitro or ex vitro conditions. Some of the examples you can see here, that is carrot, alfalfa, banana, cardamom, eucalyptus. That is, in case of alfalfa, the somatic embryos are available as artificial seed. In case of banana and cardamom, shoot buds or shoot tips are available. In case of eucalyptus, axillary buds are available. And these synthetic seeds, they are classified into two categories, that is desiccated seeds and the hydrated seeds. Again, this desiccated and hydrated seeds, they are classified into two categories. that is encapsulated and uncoated. So what do you mean by this desiccated synthetic seed? They are produced from the somatic embryos, either naked or encapsulated in polyoxyethylene glycol, followed by their desiccation. That means the somatic embryos are taken and they are desiccated. That means the water content is removed. Sometimes direct somatic embryos are removed from the moisture content, or sometimes before desiccation it is encapsulated in polyoxyethylene glycol. The examples are wheat and soybean, that is uncoated desiccated synthetic seeds are available. In case of carrot, encapsulated desiccated synthetic seeds are available. And next one is the hydrated synthetic seed. Here the seeds are also produced from somatic embryos which are highly sensitive to desiccation. So how these hydrated seeds are produced? That means here the moisture content is not removed. Why? Because when you remove the moisture content, the seeds are highly sensitive for the various process you are doing it to remove the moisture. So here, that's why these hydrated seeds are produced by encapsulating the somatic embryos or somatic propagules in hydrogel. Okay, directly these cells are covered by some of the hydrogels. Here examples: carrot, tomato, uncoated hydrogels are produced. Mango, alfalfa, encapsulated hydrogels are produced. So that is the different types of these synthetic seeds, that is the desiccated one and hydrated seeds. What are the applications of this synthetic seeds? Micro propagation can be brought about through these artificial seeds, and you can develop the plants for breeding purpose, and you can also propagate the plants by using the synthetic seeds for which the true seeds are not used or not available for the multiplication. And in some cases, when the true seeds are very highly expensive, the synthetic seeds can be done. Or sometimes the hybrid plants or vegetative propagated plants, where they are very much prone to infections which are developed by the true seeds, can be done by this method. And also you can transplant the improved or selected material in the forestry to reduce the cost of breeding, especially the European larch and white spruce trees. When you are transplanting into the forest region, the cost of breeding is very, very high. In that conditions, the synthetic seeds of that varieties can be made and it can be transplanted into the forest areas. And the conservation of germ plasm of endangered species can be done. So these are all the applications of the synthetic seeds. And next one, the protoplast culture or somatic hybridization, how the culture can be stored for a longer time. That we will deal when we are seeing the protoplast culture in detail. And also the applications of hairy root culture, that also we will see it later. And the next application is Creo Preservation. Creo Preservation is nothing but the cell or the tissue or the organ can be preserved in nitrogen. Why you want to preserve the cells or tissues or organs in nitrogen? Because if you want to preserve the cell for a longer time without affecting its original traits, that can be preserved in liquid nitrogen. The activity of the cell or a tissue or organ can be maintained for a longer time by preserving it in liquid nitrogen. So this Creo preservation is nothing but it is a non-lethal storage of biological material at low temperature. That means not only the low temperature, ultra low temperature. That means it is somewhere around minus 196 degrees Celsius. Okay. Here the temperature of liquid nitrogen is minus 196 degrees centigrade. So here what happens, all the metabolic activities of the cells are stopped and the sample can be stored for a longer time without any loss in its characters. And in some of the components or the biological material, this cannot be done because the cell viability is lost. And this Creo preservation is useful for few endangered medicinal plants. That means, you know, sometimes the medicinal plants are decreasing in their number because of so many changes in the climatic conditions and other conditions. They are called as endangered plants. So that plants can be preserved by this Creo preservation. Examples, Dioscorea chirata, Podophyllum, etc. can be preserved by Creo preservation. And next one, you can, the next application of this plant tissue culture is tracing of the biosynthetic pathways of secondary metabolites. That is nothing but, you have studied about all the secondary metabolites. That is nothing. What are the different secondary metabolites? Maybe your alkaloids or glycosides, flavonoids, volatile oils, terpenoids, tannins, resins, all those things. And each and every component is formed in the plant from one starting material through many steps into the final product. For example, if you say your receptor, it is an indole alkaloid. So first, the starting material is your tryptophan. From tryptophan, with many of the reactions, finally you get your compound called as your reserpine. Or you can take the example of atropine. It is an tropine alkaloid. The starting material is ornithine. So from ornithine, there are so many steps involved. The different products are formed, and finally you get the atropine alkaloid. Okay. So this series of reactions are called as your biosynthetic pathway. So this biosynthetic pathway can be studied or traced out by this plant tissue culture. That is how you can do it, by using a labeled precursor. That means in the ornithine, any compound or any element can be labeled, and that can be incorporated into the tissue culture medium, so that what are all the reaction taking place, how it is going on, that can be done at the final product. And next application is generation of novel compounds from the plant. That is, the novel compound can be obtained and improvement in its yield also can be done. And then as I already told you, the various functions of the plant tissues and organs can be studied by this plant tissue culture. The various functions like respiration or any other metabolic functions in the plant can be studied by this plant tissue culture. And the plant improvement can be done by the plant tissue culture by studying the diseases of the plant, and then we can eliminate the unwanted traits, and the wanted traits can be retained to produce the plants with a disease-free character by plant tissue culture. And next, the other genetic engineering methods like your mutation by mutation and selection of the mutant cell also can be done to develop a new plant with a new genetic variability. Through plant tissue culture, you can employ the genetic engineering method like mutation or selection of the mutant cell, by which the new plant with a new genetic variability can be obtained. And the last one is the production of economical valuable chemicals by this method, which are not having the normal chemical methods for its synthesis. That means the compounds which cannot be able to synthesize by the normal chemical methods can be produced by this plant tissue culture method. Okay, so these are all the various applications of plant tissue culture. So in some of the things I had given the elaborate informations, so you can make your content concise based upon the marks. If the applications of plant tissue culture alone is asked, you have to explain in detail with all the points, or otherwise you can concise your answer according to the marks and time. Okay, thank you.